Good afternoon, and a big thanks to the organizers for allowing us to share some of our work in this special session on single cell and spatial data analysis. My name is Ricardo Ramirez, and I'm a PhD student in the group of Julio César Rodriguez at the University of Heidelberg. Today, I will present our latest study on human myocardial infarction using single cell and spatial technologies. Cardiovascular diseases, including myocardial infarction, are the leading cause of mortality worldwide. After MI, inflammatory and reparative responses trigger a widespread myocardial remodeling that affects cardiac function. To fully understand the disease processes, it is necessary to describe the heart-specific intra and intercellular signaling mechanisms that coordinate this remodeling. For this reason, in collaboration with the groups of Rafael Kraman and Ivan Costa from the University of Aachen, we analyzed 8 left ventricle specimens of 5 patients that comprise different physiological zones and time points of human myocardial infarction and human controlled myocardium. For each tissue section, we generated a slide for spatial transcriptomics, and from an adjacent region, we isolated nuclei for single nuc RNA-seq and single nuc ATAC-seq. The tissue samples were provided by the group of Hendrik Milting in Bad Oeynhausen. A preprint describing our analysis is already available in bioRxiv. The computational work was motivated by 2 main objectives. First, we wanted to increase the resolution of 10x Visium, which profiles the transcriptome of spatially resolved mini-bulk samples of cell communities rather than single cells. And second, we wanted to explore spatial dependencies between cell types and molecular processes to generate descriptions of the tissue organization during disease. To increase the resolution of spatial transcriptomics, we not only located specific cell types in space, but we also described the cellular processes occurring in that specific location. To locate cell types in each tissue slide, we use available deconvolution tools such as cell-to-location or Spotlight that estimate in a given location the abundances or proportions of cell types described in a reference single-cell atlas built from single-cell RNA-seq. In our case, we built a single nuclei heart atlas analyzing and integrating our single nuc RNA-seq and single nuc ATAC-seq datasets. Our analysis recovered major cell types that conform the muscle, vasculature, and scar structures in the heart. We relied mostly on the frameworks provided by Soirad, Harmony, and Archer to achieve this. Additionally, we performed iterative integration tasks to define different functional stages of each cell type that were consistent both in single nuc RNA-seq and single nuc ATAC data. Here, I am showing a sample from a patient in a chronic condition with fibrosis, where a scar is located in the middle. After the convolution, we were able to estimate the proportions of each major cell type for each spot. For practical purposes, I only show the proportions of fibroblasts, the main drivers of the scarring process. In the bottom panels, I show the complementary information about cell states, in which each spot receives a score for each cell state. This type of mapping allows us not only to locate cell types in the tissue, but also inspect their functional variability in space. To complement the information about cell composition in each slide, we also extracted mechanistic knowledge from each spot. For this task, we employed our tools to estimate signaling pathway and transcription factor activities from transcriptomics data, Progeny and Dorothea respectively. Both methods rely on the idea that gene expression can be understood as a consequence of a cellular process. Hence, we use it as a footprint to estimate these activities. All of our tools are available in Bioconductor and Python. The application of these methods in spatial transcriptomics allowed us to look at the spatially resolved mechanistic processes. Going back to the previous example of the fibrotic patient, now we can complement information of cell composition with signaling and TF activities. In this case, I show the activity of TGF-beta, one of the hallmarks of fibrosis, an effector TF, E2F4, and the downstream target genes. This type of analysis can help us to link the structure and functions of tissues. Up to this point, we have not used the spatial information encoded in 10x Visium, which can help us to explore the tissue architecture. On the one hand, one could estimate spatially variable markers with the help of tools such as Spatially, Spark, or Transic. On the other, one could explicitly model intercellular interactions by estimating the dependencies between markers in different spatial contexts. For the latter task, Jovan Tanevsky, a postdoc in our group, developed MISTy. MISTy is an explainable machine learning framework that facilitates understanding of marker interactions by profiling their intra and intercellular relationships. MISTy allows you to build models to describe different spatial contexts. For example, one could be the interaction of markers within a spot, here referred as an intracellular interaction, or the interaction between immediate neighbors, a juxtacrine interaction. For each of these contexts, MISTy builds a component in the model called a view. It is important to note that these views can be defined by the user to answer their specific questions and are not limited to the ones I previously defined. Each MISTy view is considered as a potential source of variability in the measured marker expressions and is then analyzed for its contribution to the total expression in a method model. Additionally, MISTy provides the specific relations between markers that can explain the contributions of each view. An R implementation called MISTy-R is available in Bioconductor, and an associated manuscript is available in bioRxiv. We use MISTy to infer the cell type dependencies in different spatial contexts from all the available samples. Each network represents a dependency map in which each edge represents if the abundance of a given cell type was predictive of another cell type. I showed three different spatial contexts: intraspot, which emulates a colocalization analysis; juxtaspot, that incorporates the information of immediate neighbors; and paraspot, that includes the information of the larger tissue structure. When looking at the intraspot relationships, we observe that the most abundant cell types, such as cardiomyocytes and fibroblasts, were the most important in explaining the rest. However, as we explore other spatial contexts, relationships associated with immune responses and vasculature emerge. This type of analysis allows for the expansion of typical colocalization analysis. A similar pipeline was built to explore the importance of the tissue composition in explaining the functional differences of a given cell type. This pipeline allowed us to ask if differences in function in a cell type were associated with the neighborhood composition and potentially cell communication. In summary, today I describe our computational framework that combines the information provided by single cell and spatial data to create a comprehensive description of tissues. Our work focuses in increasing the resolution of spatial transcriptomics data by incorporating compositional and functional information. MISTy is a flexible framework to analyze spatial data that aims at dissecting interactions between markers in different spatial contexts. In combination, these analyses try to connect mapping strategies with functional interpretations. I would like to finish this presentation acknowledging all the scientists involved in this project. As mentioned before, this was a close collaboration with the groups of Rafael Kramer and Ivan Costa in the University of Aachen and Henrik Milting in Bad Oeynhausen. I would like to especially mention Christoph Kupe, Shijian Li, Monika Hanani, and Jovan Tanevsky, who work in different sections of today's work. With that, I am happy to answer any question. Thank you very much.